So this energy is released during the transfer of the electrons to oxygen through a system of series of steps called as the electron transport system. This electron transport system is include continuous is reduced coenzyme and oxidized NADH plus FADH2 are only temporarily stored in production of the electrons. The oxidation of these reduced NAD and FAD occurs by the transfer of electrons from these two R2 molecules via the system of mitochondrial enzymes and electron carriers are there. This process of the continuous electron transport system is referred to as a respiratory chain. It is also called as the activated to phosphorylation. Means ATP's production, that's why oxidative phosphorylation we call as that. The transfer of the electron from NADH and FAD to molecules oxygen involve the series of electron carriers which are arranged in the inner membrane of the mitochondria. So this is some components of electron transport system. In the complex one is known as NAD, ubiquinone oxidoreductase. It is the prosthetic group is the flavonine mononucleotide, and complex 2 is referred as a succinate ubiquinone oxidoreductase. And another complex is the cytochrome reductase. The cytochrome B and cytochrome C, they are the important components in this one. And complex 4 is called as the cytochrome oxidate. This is the cytochrome complex 4, the cytochrome A and cytochrome A3 is present in this one. The electron from the NADH enter into the electron transport chain through the complex 1. The complex convert into electrons from NADH to ubiquinone and reduced into the ubiquinol. Similarly, the electrons are released in the oxidation of the succinate reaches to the ubiquinone through the mediant of the complex 2. In the next stage, the electrons from the here reduced by the cytochrome C and transferred to the next step, that is the complex 4. In this process, the cytochrome C is oxidized. The final step, the electrons from the complex 4 are converted into molecular oxygen and oxygen is reduced. The electrons from NADH molecules which are formed in the cytosol during the glycolysis are directly transferred to the ubiquinone and the external NADH dehydrogenase, which pays the inner membrane space of the mitochondria. This is the electron transport. The production of the ATP. So production of the ATP here from ADP into two different stages are there. That is the transporylation and oxidative phosphorylation. The transporylation reactions is the phosphate group of one molecule, one already the phosphorylized compound is transferred to the ADP. So that is the phosphorylic acid from 1,3-bisphosphoric acid and 2 in the conversion of the phosphoenol pyruvic acid to pyruvic acid. This is so in the ADP, GTP plus ADP is converted into ATP plus GDP. This is the from 10 molecules of NADH plus hydrogen, the total 28 molecules of ATP are formed. For each molecule of FADH2, the molecules of ATP are formed. So, 2 FADH formed in the Krebs cycle a total of 4 ATP molecules are formed. So that the total 32 ATP molecules are formed in the respiratory chain. This is also to maintain the 4 ATP molecules are formed at the end of the Krebs cycle, means 2 ATP is glycolysis and 2 ATP in Krebs cycle. So that is the energetic and the anaerobic respiration, means in the glycolysis 4 ATPs are formed and 2 ATPs are consumed. So that's why the net gain here 2 ATPs are, and 2 NADPH plus hydrogen are formed into 2 ATPs. From that oxidative decarboxylation of the pyruvic acid, here 2 NADPH released, that is the into 2 into 3 molecules of ATP formed. So total 6 ATPs are formed. So in Krebs cycles, from 2 to 3 means 6 NADH plus hydrogen ions and 2 FADH, and from 2 GTPs are released. Totally 32 ATPs are released. So another the fermentation, another oxidative here, fermentation. The term fermentation, they are loosely applied in several processes carried some microorganisms such as bacteria, yeast, and certain fungi also. Alcohol fermentation, we know that one: citric acid fermentation, oxalic acid fermentation, lactic acid fermentation. The alcoholic fermentation is the type of respiration is carried by the yeast cells. In this process, the alcohol and carbon dioxide are produced. This is the Poischer is the first time to discover the alcoholic fermentation in the anaerobic respiration. And Buchner is discovered the enzyme zymase extract from the yeast cell, which is catalyzed process. And here the first step, the pyruvic acid is the carboxylated into the acetaldehyde and carbon dioxide in the presence of enzyme pyruvic acid decarboxylase. In the second step, the acetaldehyde is reduced into ethyl alcohol by the reducing agent NADH plus protons is the reaction to the catalyzed by the enzyme alcohol dehydrogenase. So acetaldehyde then reduced into the ethyl alcohol CH3H2OH. Another one is the lactic acid fermentation. A different course of the following in lactic acid bacillus, that is the animal tissues and some plant cells. This is the process of the lactic acid is produced instead of the alcohol and carbon dioxide. This process is the best known for the source of affecting on the milk. The pyruvic acid is the molecule formed in the glycolysis or directly reduced by NADH plus hydrogen ions to lactic acid. This reaction is the lactic acid dehydrogenase enzyme is involved. So this is the respiration system, means on the aerobic respiration and anaerobic respiration. So another one is the nitrogen metabolism and the nitrogen fixation. What is the nitrogen metabolism? So in the nitrogen metabolism, the nitrogen is the one most important element in all organisms, which is constitute the amino acids, proteins, enzymes, and nucleic acid, chlorophylls, some alkaloids also. The plant contain 1 to 3 nitrogen, their dry weight. So this is the around the atmosphere contain 98% of the nitrogen is present in our atmosphere. The plants absorb the nitrogen from the soil mainly in the form of the nitrates and ammonium. Although the main source of the nitrogen is the nitrate, but the metabolic activity of this nitrogen is ammonia. So the many plants have reduced nitrate to ammonia form from unable to synthesize the various nitrogenous molecule involved. The various biochemical processes are there. So the nitrate reductase takes place in the roots as well as in the leaves. The reduction is nitrate is the fundamental physiological process in which is the oxidized from of the nitrogen is converted to the ammonia. The reduction of the nitrate to ammonia is mediated in involved the two enzymes are there, that is the nitrate reductase and the nitrate reductase. Nitrite reductase and nitrate reductase. Two enzymes are involved. The ammonia thus the formed in the term combination with the carbon skeleton in the different nitrogenous compounds also. Nitrate reductase is the common enzyme found in the both prokaryotes and eukaryotes. This nitrate reductase is then include the enzyme. So this is the nitrate reductase is monomeric and polypeptide with the molecular weight is about 60 kilodalton is there, and then it is converted into the ammonia. Assimilation of the ammonia. The resulting ammonia is rapidly assimilated into the various nitrogenous compounds such as the ammonia, amino acids, and proteins and nucleic acid. The amino acids contain both an ammonia and carboxylic acid groups are there. So synthesis of ammonia acids, they are the reductive ammonia and transamination. So the reductive amination, the ammonia nitrogen is incorporated into alpha ketoglucaric acid, and an intermediate compound is the kryptide. That is the this is called as the reductive amination, and reduced power is derived from the NADPA. The enzyme glutamate dehydrogenase catalyzes this reaction. This pathway operates in the bacteria, fungi, and some green plants. But the recent studies, the green plants using the radioactive isotope, that is the ammonia, have released glutamate gets labeled glutamic acid. So another one is the transamination. These transfer of the amino group from one amino acid to the carboxyl group, that is the keto acid, is called as a transamination. Transamination. These enzymes involved in the transamination is the reactions known as amino transfers, and this aspartic acid is found by the catalytic activity of the aspartate aminotransferase in the some steps are there, that is the synthesis of amides. So glutamic acid then converted into the aspartic acid with help of the peroxidamase phosphate, and the synthesis of amides. These amides are additional product of the amino acids and ammonia. These assimilation of the ammonia and nitrogen involved in the synthesis of amides. The reaction is dependent of ATP in plant tissues as glutamate and asparagine are the two amides which are most frequently present. So glutamate synthesis was already in the GOGAT system, GS GOGAT system. They are interlinked. So another one is the nitrogen fixation. Nitrogen fixation. Nitrogen is one of the essential Micronutrient is required by the plant in large amount and is obtained mostly from the soil we discussed before. So, in the nitrogen fixation by chemical and physical or biological fixations are there. That is, the chemical fixation means industrial fixation. A wide variety of nitrogen chemical manufacturers are, according to the recent estimate, around 80 metric tons of nitrogen fixed in the chemical by industrial method. These methods In this method, is require high pressure and temperature of the result of nitrogen fixation takes place by the Ebersberg method. Physical nitrogen fixation. So physical nitrogen fixation means in the physical nitrogen fixation here, and the 200 million metric tons of the nitrogen fixed annually by the natural process. About 10 of these due to the physical process such as the lightning strings and thunderstorms. So lighting is associated with the water vapors by the hydrogen and oxygen atoms, and hydroxyl radicals are there. So the free atoms and radial, which are extremely reactive and combined with the nitrogen present in the atmosphere and to produce the effect of the form the nitrogen. The gaseous nitrogen and oxygen combines to form the nitric oxide. And the nitric oxide is further oxidized to nitrogen peroxide. The nitrogen peroxide combines with the water during the rains and produce the nitric acid. So the traces of this nitric acid also produced in the stratosphere by the photochemical reaction between the ozone and the nitrogen oxide. The acids are brought down to the earth along the rain, where is the react with the alkaline gradient and produce the nitrates and the nitrites. So another one is the biological nitrogen fixation. The transformation of the free insert nitrogen of the atmosphere into the fixed in the form of the soil microorganisms known as the biological nitrogen fixation. The nitrogen fixation in the soil is carried by the both free living and symbiotic microorganisms. They are called as a diatroph and wider spreads they are occurring. Some asymbiotic bacteria are there, that is the aerobic bacteria, azotobacter, and microaerobic as azospirillum. Facultative bacteria is the Klebsiella. Anaerobic nitrogen fixing bacteria, non-photosynthetic, that is the Clostridium, and photosynthetic bacteria is the Rhodospirillum, and chemotrophic anaerobic bacteria is the Thiobacillus. So some cyanobacteria also to fix the nitrogen, that is the unicellular, that is the Aphanothece and filamentous heterotrichus, that is the Anabinia nostac, and then the filamentous non-heterocyst blue-green algae also there, fixed in the Ascleroria, and there are some symbiotic association involved in the nitrogen fixation, that is the legume plants. Some legume symbiosis relation is there, that is the Phaseolus vulgaris in the rhizobium, Pseudium sativum, that is the rhizobium is also involved. Some non-legume symbiosis also there, that is the fern with association azolla and anabina corallide leaves also there. Corallide roots also anabina and nostoc, some lichens also, certain fungi and nostoc is also involved. Some cyanobacteria means a blue-green algae member. Around 40 species of the cyanobacteria belong to the families Nastacase, Cytonomaceae, Ribuliaceae, Trigonometacea to fix the nitrogen. The blue-green algae as the predominant agent which improve the nitrogen supplies, especially in the rice field in the tropics. So brought in the heterocystis bear the cyanobacterium to fix the nitrogen. So some non-heterocystis membrane like Acidoria and Formidium and unicellular is the Gliocarpa and Crococos, they also fix the nitrogen. The Anabina, they are floating per nozzle is about 30 kg of the nitrogen per hectare in the year. A symbiotic bacteria, that is the Azotobacteria and Beijering, they are important aerobic bacteria which are the capable of the fixed molecular nitrogen. The Azotobacter is the most investigated bacterium and found in the almost of the all sites. The Azospirillum, the microaerobic bacteria mainly associated with the rhizosphere of the cereals and foliage grasses. And then the Clastridium is also involved. So next one, the symbiotic nitrogen fixation. The symbiotic nitrogen fix, the organisms contribute more nitrogen than the symbiotic fixation. So there are the two types of the symbiotic association. Well, the nodular legumes and non-legume plants and the cyanobacterial symbiosis. So legume rhizobia symbiosis. The legume rhizobia symbiosis is important from both ecological and agricultural point of view. So particular the species rhizobium is effect on many species of the legume plants. That means the rhizophyllum trifolia is from the effect only with the white clover. In the initial process of infecting the roots or formation of the nodule of the rhizobia and to cluster around the root hair. This recognizes the rhizobia. The lectins may promote the aggregation of the rhizobia at the infected zone and also stimulate the mitotic activity in the nodule primordium. It is the response to the bacteria. Root hairs curl at their tips. The bacteria then invade the root hairs and the plant produces and the infection thread. The bacteria proliferate and pass through the infection thread into the inner cortical cells and they give rise to the nodules. The rhizobium, the signal molecule that play the key role in the induction of the nodulation are known as the nod factor. These nod factors are the lipocyto oligosaccharides. The genes are involved in the synthesis of nod factors are called as the nod genes. The nodule is highly organized structure. The vascular connection to the roots, the cells of these nodules are polyploid. During the nodules development, bacteria are transferred into the bacterioids, which are several times larger than the rhizobia and divided into cell wall and cell divisions are occurred. This transformation of the bacteria to bacterioids is closely related to the synthesis of hemoglobin and nitrogenase and other molecules required for the nitrogen fixation. Okay. The hemoglobin is the pink color compound and is more or less similar to mammalian hemoglobin except here amino acid. So here the functioning nodule and inner gates of nitrogen fixation. The balance between the host plants and bacterioids is delicate. There are the three factors are the direct supply of the photosynthesis, end products to the bacterioids, maintain the low oxygen concentration of the production of the nitrogenase, rapid export of the ammonia. The sucrose is delivered by the phloem into the nodule with the help of carbon and nitrogen. Metabolism is adapted to oxygen limited environment. The carbon cost of the nitrogen fixation is very host species and bacterial strain. It is suggested that 12 grams of carbon is needed to convert 1 gram of nitrogen to ammonia. And the nitrogen fixation mechanism. The nitrogen fixation was confirmed to intact the microorganisms. So here the nitrogenase is consists two different proteins are there. That is the large molybdenum iron protein contain 2 molybdenum atoms, 30 iron atoms, and 30 acid label sulfur atoms are there. So that is the iron proteins is the dimer of the identical subunits as the molecular weight around the 35 kilodalton, known as the dinitrate reductase. The nitrogenase catalyzes the ATP dependent 6 electron reduction of the dinitrogenase to ammonia. The nitrogenase activity mainly depend on the energy in the form of the ATP and reduced equivalent. The nitrogenase has the absolute requirement for the ATP. The nitrogen is not only reduced in nitrogen but also reduced in the variety of substances like acetylene and protons, nitrous oxides, and azide. Okay. So the nitrogen is return energy point of view 25 energy is lost into atmosphere. The hydrogenase is another important enzyme to involve in nitrogen fixation. So hydrogenase is two types are there: uptake hydrogenase and reversible hydrogenase. The uptake hydrogenase receive the hydrogen from during the nitrogen fixation and convert into water and produce the ATP through the oxyhydrogenase reaction. Hence, these hydrogenases protect the nitrogenase from the oxygen and provide the energy is required for the nitrogen fixation. So next one is the nitrogen cycle. This one. In the nitrogen cycle, the amount of molecule of the nitrogen removed from the atmosphere by the microorganism by the biological nitrogen fixation is returned to the atmosphere by microorganism that convert into organic nitrogen to nitrogenous gas. The transformation of these nitrogen, whether the organic or inorganic and volatile compounds, in the different ways are there. There are the three main phases. In the first phase, the atmospheric nitrogen is reduced to ammonia. The fixation may be either biological or non-biological process. The next result of these, the process of addition is the nitrogenous gas compound to soil and water. In the another phase, these nitrogenous compounds are assimilated by the plants and microorganisms. Within the soil, the ammonia and nitrate are reversible by interchanged by the action of the different organisms. In the final stage, microbes are reduced the nitrate to ammonia occurs and this is called as the denitrification. The plants are assimilated the ammonia produced in the biological nitrogen fixation into amino acids. The proteins and other nitrogenous products are released. So the bulk of these nitrogenous material added to the soil in the form of the organic nitrogen is not available to the higher plant. So the decomposition of the organic nitrogen compounds into the simpler ones brought to about the soil bacteria and certain fungi. The conversion of the organic nitrogen to the inorganic state is known as the mineralization, which result in the generation of the ammonia and nitrate. These two are the distinct microbial processes. The mineralization of the resulting the formation of the ammonia is known as the ammonification, and the oxidation of the ammonia to nitrate to the known as the nitrification. The ammonification soil is the microorganisms convert the ammonium nitrate to ammonia. The ammonia react with the carbon dioxide and water present in the soil, form the ammonium salt. The nitrification of these chemotrophic bacteria and nitrosomonas obtain the energy by oxidizing the ammonia of the ammonium salt to nitrite. The bacteria Nitrobacter further oxidize the nitrate to nitrate. Higher plants utilize the nitrate more rapidly than the form of the nitrogen. The denitrification process, the fixed the nitrogen is converted to gaseous nitrogen by in the group of the soil, that known as the denitrifying bacteria. The nitrate and nitrite are reduced in the presence of molecular nitrogen escape to the atmosphere. These denitrifying bacteria having the ability to use the oxygen from the air and hydrogen acceptors. They are the aerobically. In addition to this is the presence of nitrate and nitrate. The conversion of the nitrate into nitrogen known as the nitrogen respiration, nitrate respiration. The nitrogen lost by the denitrification may be about 200 million metric tons. This is the thiobacillus, and denitrification is the chemotrophic bacteria, which is the soil known to the oxidized sulfur or reduced nitrogen reductase. The biological nitrogen cycle is involved in the fixation of the atmosphere and nitrogen and assimilation of the fixed nitrogen by the plant. It forms the part of the food for animal, and the removal of the nitrogen from soil by the denitrifying bacteria is present in the atmosphere. This is the cycle. So, చూసారా ఏం చూసారా లంచ్ బ్రేకా